Hello, everyone. We're going to continue our unit on evolution and speciation by talking more about actual speciation now and how we get new species. So in an earlier lecture, and we looked at the movie where we looked at what is evolution and how does evolution explain the changing of species over time. So remember, there was a time where we thought that species didn't change over time, that to get a new species, an old one might have died out and then a new one came into being in some way or another, right? Well, now we know through the theory of evolution, which is backed up by a lot of evidence showing that species can indeed change over time. Okay? And what we're going to look at today is the mechanism. What is required for species to change over time? What is required for, while natural selection is acting on a population of individuals and some traits are more advantageous to others, how does that actually translate into a new species? Okay? So before we jump into that, first we need to look at what is a species. Let's put a definition on that. So here's a definition. A working definition we can use. A species is a group of organisms that maintains a distinctive set of attributes in nature. So this would imply that if a set of attributes began to change over time to a point where it's significant enough that they're different from the original population, then we would classify it as a new species. So what's changing? What's acting on it are these two different concepts, microevolution, which will then drive macroevolution. So microevolution refers to the changes in the allele frequencies in a population. So remember, we talked about allele frequencies in the beginning of the semester when we looked at population genetics. And we said that individuals have different alleles for their genes, right? That's where the variation between individuals come from. And when individuals mate, right, those allele frequencies can change over time because you're mixing up the combination of allele frequencies that can occur. Okay? And that new combination of allele frequencies can cause an individual to develop a trait, to have a trait that might confer better survivability in their environment, give them an advantage in their environment. And so if it does, they'll have more offspring, and that offspring will receive that trait, and that trait will get passed on again by more and more of the offspring who are surviving because of that trait. And that could eventually change the allele frequency in the population. Okay? We also looked at, remember, genetic drift. Genetic drift, because of mutation that occurs in individuals, you get gene mutations, that can cause different alleles to occur. Okay? And so over time with genetic drift, the allele frequencies in a population can change. But again, it's about the extent of how much it changes before it drives what we call macroevolution. Before I get into macroevolution, just remember gene flow was the term we used when individuals from different populations are able to mate with one another and they're exchanging their alleles with different populations. So then they might have similar allele frequencies because of that gene flow. They're bringing the genes to each other's populations. Okay, so again, if there are enough changes in the allele frequencies that occur over time, right, then you might get macroevolution. And macroevolution is that evolutionary change that creates a new species, and sometimes it can change groups of species depending on if several groups kind of separated from the original population into different environments. Okay, so again, macroevolution is really the accumulation of the microevolutionary changes. So microevolutionary changes may not result in a new species, but if there are enough of those, then you could get a macroevolutionary chain, which results in a new species. Okay, so the known number of species on our planet is about 1.7 million. And the article I looked up to find this mentioned that this 1.7 million are basically from our eukaryotic species, animals, especially insects, plants, and it includes protists. The article said algae, but it was more of a popular article. And a lot of times people say algae when they mean protists. So I can't imagine they're only including algae, but I put it in parentheses. So remember in a previous lecture, I think it's on Canvas, I said the estimate of the number of species, of eukaryotic species on Earth is about 8.7 million. So the difference between these numbers is this number, 1.7, is what has actually been identified. Okay, so there are a lot of species out there that we haven't identified yet. And I just realized I need to update the second bullet on here, because with that new estimate, which is getting a lot of support for, that means we have, what, about 7 million unidentified species, right? One of the problems, though, is that it's difficult to identify what an actual species is, right? So a single species may exist in two distinct populations that are in the slow process of evolving into two or more different species. That's hard for us to see that, right? So the amount of separation of time for the two populations is important. So if it's a short amount of time, then they're likely to be similar and considered the same species. But the more time that goes on, then they're more likely to show many differences and distinct differences. And so then we would call them two different species. Okay. We may find situations where the differences are apparent, but it's difficult to decide if the two populations are truly different species, right? So sometimes when that happens, we use a classification we call a subspecies, right? And so like giraffes, I believe right now we consider there's one species of giraffe, but there's about 9 different subspecies. Okay, and a little bit later, this lecture will let us see why that might be true. Okay. So, what is a species? Okay, we need a definition of what is a species. We actually need different ways to consider different species because of the diversity of characteristics, especially like in bacteria and insects, they have such a large diversity. So because of that, we have different, what we call species concepts, different ways of identifying or determining what a species is. Okay, so these 3 ways, we've got the morphological species concept, the phylogenetic species concept, also called, sometimes you'll see in some sources, it's called the evolutionary species concept. And then we have the biological species concept. Okay, so let's look at what each one of these are. So the morphological species concept, this is based on, morphology means physical attributes. So this is based on structural or physical differences between organisms. This was actually used by Carl Linnaeus. This is a guy, he's considered the father of taxonomy. Okay, so this is the person who really started to organize our thinking about biological organisms. Okay, so he classified plants, and mostly he started with plants, but he also classified other organisms based on just visible structural differences, which is what the morphological species concept is, right? So problem though is that sometimes structural differences alone aren't enough to distinguish between a species. So if you look at these pictures here, the 2 on the top, these 2 frogs look very different from one another. So if you were walking along in a forest and you came across these 2 frogs and somebody said, how many species of frogs did you see? You might say 2, because these 2 look very different from one another. Okay, the 2 on the bottom, these 2 look very similar to one another, right? Same color pattern, same kind of ridge down the side, very similar to one another. Well, it turns out, in fact, I can do that, these 2 poison dart frogs are the same species. They just have a different color pattern from one another, right? Remember, poison dart frogs, they want to advertise their poison. And so for whatever reason, they got a different color pattern, probably inherited that from their parents. However, there aren't many other traits that are different, so we wouldn't consider them a different species. right? Whereas these two are leopard frogs. This guy over here is a northern leopard frog, and this guy over here is a southern leopard frog. So they probably speciated from the same ancestor, but they were separated, their populations became separated over time, that there were several distinct differences and subtle differences that occurred. So if we really studied and looked closely at these frogs, we would probably see some definite structural differences between them. But it's hard at first glance to see what that is. So the morphological species concept, again, structural differences alone may not be enough to distinguish between the species. So we've got other ways of defining what a species is. So the next one was the phylogenetic species concept, again, also known as the evolutionary species concept. And this is when individuals have a unique combination of traits, right? Historically, we would use their anatomical structure again. But today we can use DNA sequences, we can use amino acid sequences, and so that really helps us. So the advantage is that this can be applied to all organisms. Okay, so with the morphological species concept, microorganisms can't be identified by structural differences. It's really too hard. Actually, with the onset of electron microscopes, we might be able to, but it's still difficult to use structural differences for microorganisms. So for this one, we can apply it to all organisms because all organisms have DNA. Okay, and it's testable by comparing the gene sequences. So that's an advantage. The drawback, though, is that you have to have an evolutionary history, right? They must have undergone evolution long enough in order for statistically significant differences in traits to emerge, right? If a real short period of time, you might not see statistically significant variation in traits. But then also, what's statistically significant? So this will have to be agreement amongst different scientists, right? Most groups just haven't been analyzed in this way yet. So the more easily it is to use DNA sequencing, we're going to see this really start to explode. And the number of species, if we use it this way, can actually double. So we could end up classifying subspecies as species with this method. There's actually a lot of scientists kind of working on gathering a lot of molecular data, DNA data from different organisms. And we have big supercomputers to hold all of this data and to work out the similarities and differences. So that's the phylogenetic species concept, right? Oh, let me mention, I didn't mention here. Here you can see, this is an example with the stickleback, right? These are called stickleback fish because of the spines they have growing out of their backs, right? So we have some that have with spines and some without spines. So this is a freshwater form without spines, although they drew them here. But the other thing is he doesn't have these plates, right? Here's an oceanic form with these lateral plates that go front to back on the entire fish, right? And so this is a trait, these spines and plates, that have been established genetically and that they are inherited, okay? And so while these fish really look very similar to one another, we can establish them as different species. Oh, I'm sorry, I was looking at the dorsal fin. This is the spine coming out the bottom here. So this guy is without a spine. Okay, and this one has a spine. And again, of course, the plates that go all the way down. Okay, so these are two, so just lacking a spine doesn't necessarily mean they're different species. But again, looking at their gene sequences of these traits, spines and plates, we can see that there's a significant amount of differences to classify these as two different species. And oftentimes what we do then is we may look at their behavior and see in the lab, can these two interact with one another? And if they ignore each other, then we would definitely call them two different species if they can't mate with one another. Okay. So we might layer on, and I'm kind of setting the stage for the next definition. But so we might layer on some of these different concepts in order to determine if it's a species. Here's the other thing though, this one's oceanic, this one's freshwater. And so if this freshwater doesn't empty into the ocean where these fish live, and they don't have any overlap, then you can see that that might be a reason for speciation to occur. Okay. The next one is the biological species concept, and this has to do with interbreeding in nature. Right? So members of one or more populations that interbreed in nature, and not only that, but once they interbreed, that they actually produce viable, fertile offspring. Viable means that the offspring live, right? And then fertile means that when those offspring reach reproductive age, they can then produce viable offspring as well. So it has to, the offspring reproduction has to perpetuate. Okay, this biological species concept was first suggested by a researcher named Ernst Mayr back in 1942. And this centers around reproductive barriers. Okay, a reproductive barrier is something that restricts individuals from exchanging genes. So I had already mentioned gene flow before. Remember, we talked about it before. If you have a gene pool, right, then all of these individuals in the gene pool are able to reproduce with one another. Okay, and so you get gene flow. So you see the different colors represent the different types of genes. And there's purple and green and red. And maybe they turn out to be those colors, right? So here within this gene pool, there's free gene flow. So all of these individuals are free to mate with one another and form. The allele frequencies won't really change too much over time. But if we added some kind of isolating barrier, okay, a reproductive barrier is one that prevents those individuals within this area from mating with this area. Now you can see we've got a reproductive barrier between those with green and purple traits with those with the red trait. So this population now can reproduce, and the red trait phenotype will continue on. And if it turns out that that's more advantageous, the gene frequency could change dramatically from this group that's reproductively isolated. Remember, reproductive isolation means there's no more gene flow between these groups. Okay, they can only mate within the groups that they have. Okay, so we most biologists, they tend, depending again on what they study, but the most broad definition, we tend to try to use the biological species concept, because again, it has more to do with can these organisms relate to one another in nature? And the ultimate relationship is through breeding. Okay, and again, even if you can relate to one another and you can produce an offspring, if that offspring though dies right away or is infertile, can't produce, then it wouldn't be considered, they wouldn't be considered the same species. Okay. So there's some problems though with this definition as well. So it may be difficult to determine if two populations are actually reproductively isolated. What we're going to see is that there's different forms of reproductive isolation. It doesn't just occur with a physical barrier. There can be other ways for reproductive isolation to occur. There are cases where two species can interbreed, but they just don't. So is that a reproductive isolating barrier, or what's going on there, right? But then also, obviously, then this concept can only apply to sexually reproducing species. Okay, if you can't apply it to asexual species. So in the ecological species concept, then a species occupies an ecological niche, right? And so that one you can apply to asexual species. And we tend to do that with bacteria. So ecological species concept, I didn't really bring it up much here, But that one we might use with bacteria because certain bacteria proliferate on a particular medium, right? They need particular resources, their ecology. And so many times we classify bacteria in that way, right? But for most of the talk on species, we'll use the biological species concept. Okay, so this is going to bring us into the reproductive isolating mechanisms. What I'll do is I'll stop the video here so I keep it short. And the next video will start with reproductive isolating mechanisms.